We're pleased to have you join us in the second CCAN Roundtable discussion of 2018. The title of today's presentation is Summary of a Workshop on Monitoring for Acidification Threats in West Coast Estuaries, San Francisco Bay Case Study. This webinar series is hosted by California Current Acidification Network, and the purpose of these roundtable discussions is to focus on ocean acidification on the West Coast and its effects on marine ecosystems. I'm Bruce Steele, a sea urchin diver and member of the CCAN Steering Committee, and I'll be monitoring today's session. We also have Shalyn Bush from NOAA online, who is running the logistics of the webinar. We're very excited to have Bill Trowbridge and Karina Nielsen speaking with us today. Bill is a program manager for the Regional Monitoring Program for Water Quality in San Francisco Bay. The RMP is a multifaceted regional monitoring program that studies ecosystem health in the Bay. Karina Nielsen is Director of Estuary and Ocean Science Center and a Professor of Biology at San Francisco State University. Karina also serves on the California Ocean Protection Council's Science Advisory Team, the Governing Council for the Central and Northern California Ocean Observing System, and the Management Advisory Board for San Francisco Bay National Estuarine Research Reserve. Today's roundtable will begin with presentations from Bill and Karina on the outcomes of the acidification workshop for San Francisco. We will then follow with any questions. Please note that during the presentation, attendees will be in the listen-only mode. You're welcome to type in questions to the presenter in the box at the bottom of your control panel on the right of your screen. We'll be monitoring questions, and we will field them at the end of the presentations. We're also recording this session, and we'll share the recording with the CCAN website after this webinar. You can also find earlier webinars, the CCAN workshops, and CCAN workshops on the CCAN website. Okay, I'll hand this over to Phil now.
Phil, we're not hearing you. This is Shalyn.
Okay. Yes. Okay. You can hear me now?
Yes.
Okay. Great. Well, good afternoon, everyone. Thank you for joining us for the webinar. My name is Philip Trowbridge. I'm at the San Francisco Estuary Institute, and I'll be doing the first part of this presentation. Hang on. I'm having a little trouble here. There we go. Okay. So we have two parts of the presentation. One is about the workshop outcomes, and that's what I'm going to cover. And my co-presenter, Karina Nielsen, will be covering our monitoring initiatives that are being undertaken as a result of that workshop as we start to develop a monitoring program for acidification in San Francisco Bay. So just to start with a few specifics about the workshop. We held this workshop back in 2016 in October, so a little over a year ago. It was held here at San Francisco Estuary Institute in Richmond, California. We had great participation. We were really excited. We had a good mix of scientists and managers to talk about this issue. And within the science community, we had people coming from the chemistry side, people coming from the biological side, and then within the manager group, we had people who were principally responsible for water quality, others who are involved with ocean policy, and still others related to fisheries. So it was a great group of people, a great organizing committee, and you can see all of the logos at the bottom of the groups that either contributed funds or contributed in-kind time to make this workshop a success. We went into the workshop with four main questions that we were trying to answer, and these came from our managers, and they were essentially asking us at the top level, is this something that we need to worry about in San Francisco Bay? We have plenty of other things to worry about and manage in the Bay, so is acidification something that needs to take away resources from other needy issues. After that, the questions of, well, if it is an issue, what are the likely pathways and where are we expecting to potentially see it? And what type of indicators should we be looking for? We can't monitor everything, so we need to think about the most sensitive chemical and biological indicators so that we could assess trends and status for acidification in this estuary. And then finally, the question of monitoring. What is the best way to do monitoring within an estuary like San Francisco Bay? And by extension, what about other California estuaries and what can we learn about that? So we had 2 days to cover these topics, a great group of people, and my job here is to present some of the findings. So at the first level, that question that our managers asked, is this something we need to worry about? The conclusion was that acidification is expected to impact estuaries like San Francisco Bay. Something like San Francisco Bay, a deep channel estuary, we can have the inputs of low oxygen, low pH water from upwelling events coming into the bay through the deep channels, especially in the outer parts of the bay. The species of most concern are shellfish species, as well as particularly those that exist within near the mouth of the estuary. The other thing we concluded was, despite having decades of monitoring and a large community of academic institutions and other agencies collecting data on San Francisco Bay, we didn't have the data for chemical and biological effects to say what was going on in San Francisco Bay at this point. We have not been taking the carbonate chemistry measurements that we should be, and we have not been connecting the dots with some of our biological monitoring. So we have our work cut out for us as we move forward. Here's just a graphic showing the bathymetry and topography around San Francisco Bay. And you can see the deep channel through the Golden Gate, which would be the pathway that we identified for intrusion of low pH water from upwelling events. So the conclusion geographically was that this area, I believe you can see my mouse here, this area in Central Bay where the deep channel is coming in through Golden Gate is the area that would be most at risk. And this also happens to be some of the areas where we have shellfish resource of concern. So moving on to question 3, which is, what are our sensitive indicators? And I think this is important for us to help focus our attention, because acidification is one of these effects that has cascading or can have cascading effects through the ecosystem. So potentially you could measure lots of things, or lots of things are related to acidification. So it's important that we try and focus our attention. One of the, as suspected, impacts to fisheries and shellfish habitat were one of our primary concerns. But I think something we didn't expect going into this was that the acidification could potentially be changing the phytoplankton community. And that's something that has relationship to some of our other efforts in San Francisco Bay as we study nutrients and the effects of nutrients on the phytoplankton community, and also the incidence of harmful algal blooms, which are important and have a public health potential consequence. So that was an area that we didn't expect, but we were very interested to see that because of its connection to our other programs. So we move into question 4, which is what type of monitoring program we should have. We took a little bit of a detour and acknowledged the fact that acidification was an issue that was going to require us to break down the silos between our different programs. That programs that had previously been quite separated, such as water quality versus restoration, really started to have the same started to have a very common interest in the question of acidification. For example, restoration of a shellfish area, a shellfish bed. We would want to know if this is an area that's potentially subject to acidified water coming in from an upwelling event, because that would reduce the likelihood of success for that restoration. On the other side, restoration of something like an eelgrass bed that can have an ameliorating effect on acidification might be looked more favorably upon if it's co-located with shellfish restoration in the same area. So before we got into the question of types of monitoring, we realized that we needed to break down the barriers between the water quality managers and the fisheries and restoration managers, and work across jurisdictions. So just for example, on this is a map showing our shellfish beds in San Francisco Bay. And you can see a number of them clustered in this area of Central Bay and Richardson Bay, where might be subject to upwelled water. And if you overlay that, mentally with where we have submerged aquatic vegetation, you can see that at least here in Richardson Bay and some of the fringing environments off of Tiburon, we are having the co-location of shellfish and submerged aquatic vegetation that would put a couple of different resources together to mutual gain. And for water quality managers, we need to start thinking about these multiple benefits. So we had some logical outcomes in terms of next steps. In the short term, we're going to continue to look at the data that we have and basically look for ways that we could piggyback onto our existing monitoring assets. We identified carbonate chemistry monitoring as our first step of monitoring. It's an obvious gap, and we think it can be piggybacked onto some of the nutrient monitoring that's already happening. However, we recognize that the interaction between the chemistry and the biology was ultimately going to be very important for giving managers information about the effects of acidification, not just the occurrence of it. And so long term, we're looking at developing this coupled chemical and biological monitoring program. So one of the goals of this workshop was not just to talk about San Francisco Bay, but to use it as a case study for other estuaries. So we took advantage of the brain power that we had in the room to try and tackle that topic. And it was a challenging one because California is a large state and has a wide variety of estuaries. So we could not go do a deep dive on any other one estuary or even too much of a comparison between them. But we did think about the process, and the process that we outlined in the report is essentially to follow the steps that we had chosen to follow for San Francisco Bay, and not because we want people to do what we did, but because we thought they were the right steps. That it makes sense in all cases to compile the existing data and assemble experts to talk about it and synthesize it as your first step. It makes sense in all cases to develop a conceptual model for the impairment pathways, thinking about where the impairments might be occurring in San Francisco Bay, like for instance in the deeper channels near the mouth and the Golden Gate, was helpful in terms of knowing where we should be focusing our attention. It makes sense in all cases to then look at your existing monitoring assets and decide how you can leverage those, and then based on your situation, the assets you have, the opportunities that come up, and the priorities within your stakeholder group, take the next step in terms of adding new monitoring or new studies. We did, well, we didn't look at every estuary in California, and that was beyond the scope of the project. We did think about a couple of different estuary types and how they might be different. And there were three that we talked about specifically. The deep ocean forest bays, such as San Francisco Bay, The narrow river mouth estuaries, something more like the Russian River, and then lagoons. So something like Morro Bay, or maybe not Morro Bay, but a lagoon with an actual closed mouth. These will all act differently, and something like the deeper San Francisco Bay with the deep channel connection to the ocean and potential for upwelling intrusions will tend to behave similarly in that you will have this acidification risk closer to the mouth and closer to the bottom. The narrow river mouth estuaries are going to be more affected by the freshwater flows and the alkalinity from the watershed, less susceptible to ocean-derived acidification. And for a lagoon, when it's fully closed, obviously the ocean effects are going to be minimal or zero, and local and internal processes are going to dominate. So we recognize that though we talk about these estuaries as types, that a single estuary can have all of these forms over the seasonal cycle depending on flows. So you have to recognize where you are in that continuum and which of these processes are most important. So this is the point where I'm going to now hand it over to Karina. I'm going to pause for a minute here, and if there were any immediate questions, we could take those, and otherwise we will move on to Karina for the second half of the talk.
Yeah, I'm not seeing any questions from the group. Folks on the line, if you do have questions, feel free to put them into the questions box. You can also raise your hand, and I'll look out for that.
Okay, then. So Karina, we'll pass it to you to finish the presentation, and we'll do questions at the end.
Great, thank you. Just a sound check. Can you hear me?
Sounds good.
I can hear you fine.
Great, thank you. So I'm Karina Nielsen, and I work at San Francisco State University. So my role here in this part of the talk is to tell you a little bit about what we're planning to do to develop a monitoring program within the San Francisco Estuary with our partners and some of the steps that have already occurred. One of the major efforts that we've been undertaking at the Estuary and Ocean Science Center together with the Central and Northern California Ocean Observing System, colleagues at UC Davis, and with the Environmental Protection Agency is to deploy a mooring array in San Francisco Bay. And I think you can see the little pin here that we have that will have a buoy and an associated.
We can't see your slides. If you could go ahead and press show your slides.
Sorry, people should show my screen. All right. How's this? Can you see it now?
Let's just give the software a second to update. Not seeing it yet.
Screen, hold on.
Okay, so yeah, I'm seeing the slide sorter if you want to go ahead and...
Okay, how about this? Can you see it now?
Yes, we can. Mm-hmm.
Okay, thank you. Sorry about that. So this is a map showing, San Francisco Bay, and there's a little pin here next to the Tiburon Peninsula and just around the corner from Richardson Bay. And that's the location where we're scheduled to deploy a carbon dioxide monitoring buoy at that will collect data on the surface waters, and a companion buoy at depth that will measure the potential incursion of OA along those deep channels. And here's some just some images. So the buoy is one that NOAA and the PMEL group has designed that measures the atmospheric concentration of CO2, the CO2 in bay water. We also have instruments attached to that buoy on the base that's inside the water for pH, salinity, water temperature, dissolved oxygen, chlorophyll, and turbidity. And then this deep mooring over here, which will be a companion, has an instrument called the CFOX on it. Some of you may be familiar with that. It'll be at about 60 feet deep in the water. It's close to as deep as it gets at that part of the bay without getting into the shipping channel, which the Coast Guard didn't want us to do for obvious reasons. And that's going to measure pH, salinity, temperature, and dissolved oxygen as well. And it will be, the program will be accompanied by discrete samples of water collected by bottles to calibrate the instruments and everything. But one of the neat things about this, and this is scheduled to go in the water actually February, about the third week in February. So I don't have any data to show you now, but we should soon, and it will be live streaming. So that'll be pretty informative. There are a couple of other, we were talking about how important the partnerships are, and we have, through the workshop and the opportunities to communicate with partners, been able to recruit in other interested scientists who are working and have different resources available to them. So as one example, Dr. Andrea Fassbender, who's a chemical oceanographer from the Monterey Bay Aquarium Research Institute, she is doing a study of the role of watersheds and how the discharged water may influence coastal alkalinity, which could potentially influence the accuracy of our measurements, and also have some buffering capacity potentially on the outer coast. And so she and I are collaborating. She's going to be taking samples together with our buoy and mooring array to understand what that contribution might or might not be from watershed. So she's working with me in San Francisco Bay. Her project has a larger scope along the coast of California. So that's one interesting development that's coming out of this workshop and emerging work network. In addition, there has been a lot of interest in the role of seagrass and kelp as a potential ocean acidification management tool. And I think some of you probably have heard that this report just came out from the Ocean Protection Council Science Advisory Team Working Group, looking at what we know so far and where we might go with this type of approach. And some of the main findings from that are that the protection and restoration of seagrass and kelp is basically a no-regrets strategy in terms of coastal management because of all the other benefits of those habitats. And the science that's been emerging to date of monitoring inside and outside of those habitats has shown that, yes, indeed, under certain conditions and times of year and day, that vegetation, that habitat can increase local pH, so ameliorate the corrosive conditions through the process of photosynthesis. And we're learning a whole lot more about where and when they may ameliorate OA, and there's a lot of restoration potential in San Francisco Bay, particularly for seagrass. So this is obviously of great interest locally. I just thought I'd show you a couple of the graphics from that report since it's just come out a few days ago. I think most folks are probably familiar with the basic concept, but the idea is that as the water passes through a bed of vegetation, be it the eelgrass here shown at the top or a kelp forest shown here at the bottom, photosynthesis would draw down some of the CO2, and that would be a way of sort of sweetening the water. There's also the potential for carbon sequestration if some of that biomass or some of the sediment that's trapped in those beds gets buried in the sediment. And with kelp forests, even though they are not in a sediment system, they're usually on rocky substrate, there is some potential for sequestration in deep sea canyons. And we don't know a lot about the magnitudes of that, but those are some of the ongoing questions that a lot of folks are trying to understand more about. Obviously, there are potential advantages of co-locating seagrass or kelp with aquaculture or with restoration of native oysters, and that may be beneficial. The sort of the where and the when and the how much benefit we might expect from this process of photosynthesis is still an ongoing topic of study. And the graph over here on the right is just showing you sort of the day and night cycle over a series of 3 24-hour cycles. And you can see that the blue line is showing you how the pH increases inside a bed of seagrass relative to outside. And that if you look in the evenings, you can see that actually the opposite is happening, that it's actually getting a little more acidic, but that overall, at least during this period of time, there is an overall average increase in the pH, and we believe that there may be some benefit to organisms of this change. So that's sort of a question that's ongoing. The work in kelps is a little bit not quite as resolved yet. We don't have the inside-outside comparison, but we're learning more and seeing similar types of patterns in those data as well. So the sort of the emerging understanding from this work that is related to the ongoing monitoring for carbonate chemistry is that we are continuing to learn more about the magnitude and the spatial extent of how ocean acidification may be ameliorated by this underwater vegetation and where it may be most likely to work. The next step is also to understand, is that slight reduction in, increase in the pH from photosynthesis, does that actually benefit, say, oysters growing in or in close proximity to that vegetation, or mussels or other shell-forming organisms? And the promising aspect is that that increase is about 1/10 of a pH unit, which is roughly equivalent to the human inputs into the ocean and the magnitude of the change that we've seen so far. So we think it's of a magnitude that may be beneficial. We are starting to do some active carbonate sampling in San Francisco Bay with USGS, and I now have to correct my slide because was supposed to start on the cruise that I believe was set for today, but we had an instrument failure, so it is going to be delayed, and we are regrouping to do that on the next USGS cruise. But Ryan Anderson, who's a student with us, will be working with Christopher Gobler from Stony Brook University, who's loaned us some instruments, and we're going to get a first look on one of these regular water quality cruises that has been as a long-term data record. So that's very exciting. We also have been planning with our colleagues at NOAA for their 2020 coastwide cruise, and plan to coordinate sampling among the buoy work and the moorings, the USGS cruises, and the NOAA cruise, so that we can put those different measurement sets, data sets together in a way that are cross-calibrated and can learn more from all those different pieces. And this is part of the array of the developing West Coast ocean acidification sort of multi-stressor observing network, and that's sort of the point, is to integrate across all these pieces, so we have a comprehensive as possible an understanding. This is just another slide showing on that 2020 cruise the way in which we are going to be overlapping and integrating those measurements. This is where the mooring and array will be. There's a USGS regular sampling station over here, and the NOAA cruise will come in and hit some of these benchmark sites, and we will sample simultaneously. So that's very exciting. We're looking forward to having that happen. And that's where we are with our monitoring program for San Francisco Bay and its development. We're looking forward to expanding it and growing that as resources and expertise becomes available. And that's the end of my presentation. I'm happy to take any questions.
I should just add, Karina, that this URL that we're listing is where you can get a full report from the workshop with all the recommendations and quite a bit of detail, way more detail than we could cover in this presentation.
Thank you.
Well, we have a question up. We only have one, so whoever would like to field that, you can take your shot at it. Phil or Karina, you can see the question.
So I'll just put that to the group. It's from Emily Joy Frost, and she says, "Hi, I'm a member of the NZOAC and CARIM in New Zealand and in charge of a CFET and microcat mooring in a highly quote-unquote impacted site in New Zealand. The question is, do you have any areas in your U.S. state which is most highly impacted by nutrient runoff and hence large algal blooms? What policy or management strategies do you have in place to attempt to resolve this impact and limit the effect on coinciding aquaculture?"
Phil, do you want to take a stab in it? Okay.
Yes, I'll start. I will. Yes. Okay, so I think there's a number of pieces to this. I guess maybe what I should say is in San Francisco Bay, we recognize that there is a link between nutrient management and acidification, and we're still trying to understand what that link is, or how I guess the exact amount of impact from the anthropogenic nutrient inputs. But that's part of the way we're looking at it, and that there is a management lever that can be pulled in terms of reducing nutrient inputs to San Francisco Bay as a way to not have, to not exacerbate the ocean acidification. We're not at that point yet. I think we're still in the study mode for it. That studies are also happening in Southern California, and our partner agency down there called SCWRP, or the Southern California Coastal Water Resources Project, is heavily involved in this question and is studying it with places like the city of Los Angeles and the city of San Diego. So I'd say there's a lot of research on this topic happening, in California and also in the states of Washington and Oregon. For our local situation, we haven't made any decisions yet or taken any regulatory actions because we still haven't fully understood the situation.
Yeah, I think the lack of data is part of the problem, but also in San Francisco Bay specifically, the dynamics of phytoplankton blooms and nutrient loading and sedimentation and light and the role of invasive clams. It's not your typical estuary. It doesn't have the typical response of a lot of other estuaries that are eutrophied. So that adds a complicating layer to understanding how the relationships might actually be mechanistically among phytoplankton, nutrients, bloom formation, and ocean acidification.
Right, and really the area where we might have the most sensitivity is the coastal ocean outside of the Golden Gate. And Andrea Fassbender's work, where she's going to start looking at the effect of the alkalinity plumes coming out of the Golden Gate, should be very helpful for us to start understanding what kind of incremental effect there is on the pH out in the coastal ocean as a result of the watershed discharges.
Right, the management lever is kind of there, and some of the sensitivities may be actually in the quality of the water that's coming out in the plume. And there's a lot of water management ongoing, obviously, in the estuary, but those are I guess that's where the management lever is and where some of the impacts may be more likely to be observed in terms of water quality coming out of the bay.
So I have two questions for you guys. This is Shalyn, and one for Karina. Where is the data from that new mooring going to sit? And then my second question, which is broader, is I'm imagining that it's a really complex environment, trying to figure out how to monitor for and respond to OA in a very large state with a lot of coastal territory. So Phil, you mentioned this briefly the beginning of your talk, but I was wondering if you could elaborate a little more on just the process that California is doing to start tackling OA.
Phil, do you want me to take that, or are you?
I think you're better qualified for both.
Yeah.
Both questions.
That's what I was thinking. So, the first part of the question was, where will the data reside from the buoy and the mooring? And that will, it will come up through the, it will live on the Senku site, as well as the laboratory site for the Estuary and Ocean Science Center. So we are collaborating with Senku's to stream the data and make them available to a broad audience. The second part about what California is doing, so through the Ocean Protection Council, there actually has been an investment in some monitoring projects that have fairly fixed duration and time at this point, but have geographic scope in different estuaries along the California shoreline. And they're looking at it in conjunction with the role of submerged aquatic vegetation. That's one piece. They're also, I think, on the entire West Coast, they're collaborating with Oregon, Washington, and possibly up in BC. I'm not sure if I'm remembering that correctly, but to do an inventory of what the monitoring assets are, what the funding situation is for those instruments and assets, and just trying to make sure that I think they're considering, well, depending on what they find, I don't know what they're going to do, but they're considering funding to sort of harden or support that network as best they can.
Great.
Does that answer your question, Shelley?
It does. It absolutely does. We have another question in from Emily Knight, and it says, you discussed future data collection and research. Is there any plan to reconvene the participants from this workshop to share new knowledge and discuss any progress in management?
So
Phil, you want to? Yeah, go ahead.
I'll go ahead. Yeah, and first off, say hello to Emily. Emily was one of the organizers of the workshop, so we're nice to reconnect with you. We don't have imminent plans, but we are. I think it will be something that we would want to do in the next few years. Perhaps as a little bit of this new data collection comes online, and we have something more specific to talk about and some details to dig into, I think it would be really good to reconvene that group. And it had also the benefit, as you know, of connecting the network of researchers on acidification in the Bay Area, and that is worth doing just in its own right. So I would say we should look to do that again in probably 2019 when we have a little bit of data available, and we could also at that point be putting the final touches on any plans for the NOAA 2020 cruise as it comes into the Golden Gate.
Yeah, so I would say I think all the participants are interested and willing to do so, but there isn't, we don't have a concrete plan yet. I might add just one more thing. There is also a concerted effort and interest in gathering the existing there's a lot of pH data monitoring that's gone on in the Bay using glass electrodes, which is a technology that just is a little challenging to put all the different instruments together and coordinate them at the accuracy precision level, perhaps that would be useful. But the hope is that we can perhaps improve on the existing network, say within the estuary that already exists. How can we add or contribute based on these anchor moorings, these high precision measurements? Are there ways to use proxy measurements to better advantage? And that's part of part of the work we're going to try and do.
Great, thanks. So we have another question in, and I'm just going to read that. So it says, hi all, I'm Aurora Reichart, a postdoc at the Bodega Marine Laboratory, investigating seagrass OA buffering capacity. We have been deploying sensors since 2014 in different seagrass meadows. However, to calculate the extent of the effects, you need seagrass maps. And as far as I know, detailed maps of seagrass meadows are not available. Is seagrass mapping among the goals of CCAN?
I'm pretty sure that within the OPC, they're very aware of that, and that is one of the action items that they are pursuing.
Great, thanks. Yeah, and this is something potentially we can discuss a little later on with others from CCAN, unless Bruce, you want to jump in there from a CCAN point of view.
Well, I think one of our long-term goals has been mapping, but I mean, to be honest, we're stuck up against the barrier of we're really not funded, so we're completely dependent on research from funded groups to do that sort of work. I mean, so that's kind of just honest evaluation where we're at.
I think we share your your concern about that. Yes.
The original goals.
But I think there are funds. I mean, the state of California is investing. You know, there are folks who are investing in this, and I think that you'll see some progress soon.
Go ahead. No, I would.
Wait, this is Diane Pleschner. She has something to say on the CCAN perspective.
No, actually, the original goal of CCAN was to develop a roadmap to develop a monitoring network coastwide, and I think we're doing a pretty good job of linking science and the fishing community and the agencies. We've got a lot of cooperation going up and down the coast now that we didn't have before the initial CCAN workshop. So, you know, the money's important, and continuing to build on the knowledge base that we're gathering, I think is important. But you know, we appreciate everybody's help in moving this forward, because it's critically important to understand the impacts that OA are going to have over time.
Another one. So I have another question, Phil. I read the overview of your workshop, and there was quite a bit of talk about trying to connect the buoys inside and outside. So can you give us some perspective of how would you look at the existing buoys that are set up in Monterey, or potentially other offshore buoys that you might be able to link with information about what kind of water is coming out of the bay and how potentially that affects open ocean conditions?
Sure. Well, I think there's one word for that, and it's modeling. You know, as we have started to expand our modeling capacities for the nutrient research, that has necessarily extended our modeling grid well offshore into outside the Gulf of Farallones and along the coast in order to characterize the inputs and outflows from the Golden Gate. That has a side benefit of helping us with understanding when and the potential for intrusions of low pH or upwelled water. So I see the model as a key glue to help us to connect monitoring that we do in the bay with offshore stations. I also just think the work that some of these researchers like Andrea are doing to you know, have their monitoring network extend from inside the estuary to the offshore environment will also help us understand that. So I think there'll be a couple of methods, but I think modeling is an important one for helping us to understand mechanisms and to connect the dots.
I agree with what Phil is saying. I might add that, you know, in our ideal world, we'd have another mooring array or buoy right outside of San Francisco Bay, in Drake's Bay, perhaps near the source of upwelled waters that would be likely to transit into the bay. And I think there are some efforts underway to try and raise some funds to do something like that. But I don't think we've been successful yet, but people are working towards that.
Yeah, I think the point, we should all look at these models that are being developed for different purposes and recognize that it is all one water mass, and you get more benefit from them than just what they were built for. We were able to use these same models to help us track the trajectories of microplastic particles leaving the Golden Gate. This was not something that we intended when we originally built the model. So it's really an investment in infrastructure for the region, and it will help many different types of studies.
Okay, I have another question. This is Bruce again. So I have a question having to do with legacy heavy metals in San Francisco. Have you guys, or was it a question in the symposium about how potentially ocean acidification is going to affect the bioavailability of some of those legacy heavy metals?
Sure, I'll handle this. We did talk about that. That was one of the ideas that we vetted. The feeling was that the change in solubility of the metals was going to be pretty slight compared to the more direct effects on calcifying organisms. It certainly was there, and we thought about it in terms of a conceptual model of this is a pathway of potential exposure. But in the realm of all the different potential risks, it did not rank at the top. I think the other thing to recognize that the most of the legacy pollution that we deal with and manage in San Francisco Bay are things like PCBs and mercury. So PCBs obviously are not going to have much of a solubility effect from a pH change of 0.1, and mercury, the bigger effect in terms of bioaccumulation is from the methylation process, which has more to do with biological activity. So when we thought about that, just kind of working our way through a risk, a semi-quantitative risk analysis, that issue did not float to the top. I mean, the more important things or the more sensitive indicators we felt were direct impacts on calcifying organisms and changes in the phytoplankton community.
Well, we don't have any more questions, so unless somebody has something they would like to bring up, we start bringing this to an end. Video recording of this and PDF of this session will be shortly available on the CCAN website. You can also join the CCAN listserv on the website, where you can find out about CCAN activities and future webinars. Thank you for attending the CCAN Ocean Acidification Roundtable today. We welcome any feedback, further questions, or suggestions for topics for this webinar series. You could submit input by emailing us at the address listed on the slide. Our next roundtable discussion will be April 2nd, and we'll focus on risks of ocean acidification in the California current food web. And Shaylin's going to be co-hosting that, so we look forward to that. I think everybody will be very interested in what the biological forecast is. And I'd like to thank Phil, and Karina, and Shaylin, and everybody who helped put this together. And that's the end of the session.